All right, ladies and gentlemen, welcome back. So today we are going to just talk about the general structure of DNA, right? So when we talk DNA, remember that this stands for deoxyribonucleic acid. This is your substance of inheritance. And that hereditary information really is encoded in this chemical language based on the nitrogenous bases and the order in which we find them. And that DNA then, using that set of sequences of nucleotides, is what sets the development of all of the different traits that exist in organisms. So when we go from DNA to RNA to protein, going through that transcription and translation process all ultimately boils back down to the original DNA code, and that DNA code is what gets copied and sent from parent to offspring. So when we look at general DNA structure, right, it is a polymer just like all of our other major macromolecules. So that means it has a monomer. And so when we look at DNA, it is a polymer of nucleotides, right? And those nucleotides consist of three major parts. You've got your phosphate group, you've got your sugar, and you have your nitrogenous base. Now, when we look at your phosphate group, this is going to be identical from DNA as well as RNA. The sugar here, because we're talking DNA, is deoxyribose. And actually, the only difference between the deoxyribose here in DNA and the ribose that we see in RNA really actually has to do with what comes off of this second carbon. So here in your deoxyribose, it's only a hydrogen. When I look at the group that comes off of that sugar in RNA, it's actually an OH, it's a hydroxyl group. So literally this is deoxygenated ribose. That's where the name comes from. So we removed an oxygen here, and that is the only difference that makes ribose different, which is pretty crazy. And then we look at your nitrogenous bases, and you have four potential options. You've got A or adenine, T or thymine, C for cytosine, and G for guanine. We go into your RNA, then uracil also exists there. And so that structure of those bases then are also a little bit different depending on which nitrogenous base we're looking at. So depending on whether we're talking about a double ringed base or a single ringed base, that's where the purines and the pyrimidines come in. This really was the struggle for Watson and Crick when they were trying to build the model of DNA, is to figure out how these rings came together. We knew their basic chemical structure, but we didn't know how they fit. So when we look at purines, those are your double ringed bases, right? So they have two carbon rings to them. And so your adenine and guanine are both purines. We look at your pyrimidines, and they are single ringed bases, so they have a single carbon ring. And one of the things that Watson and Crick were trying to struggle with is, how do I fit them together? Well, once they got that photo 51 image from Rosalind Franklin's work, they realized that this ladder had to be parallel. That meant that the distance from one to the next had to be identical from every rung. And so when you put two purines together, it's too wide. You put two pyrimidines together, it's too narrow. The only way to make this work is to have three rings across, a purine with a pyrimidine. So when we look at ratios of bases in your DNA molecule, there's a reason that adenine and thymine always equal the same, that guanine and cytosine always equal the same. It's because they are able to base pair with each other in order to properly meet the data from that X-ray data to keep this the same width all the way through. And the reason that it works has to do with their hydrogen bonding. All right, so adenine and thymine are only capable of making two hydrogen bonds, while guanine and cytosine are capable of making three. And so to increase the stability of that molecule, they want to make as many hydrogen bonds as possible, which is why guanine and cytosine are going to end up matching up, because they can make three. So while guanine could make only two hydrogen bonds with thymine's capacity, it wants to make three. It's more stable to make three. And so it seeks out that cytosine instead. And that's why they kind of have evolved the way that they do based on their way their chemistry works. So when we look at DNA, right, you get those nucleotides that are held together by those hydrogen bonds, right? Once again, G with C because they can make 3 hydrogen bonds, A with T because they can make 2, and that's where those nucleotide bases come from. And this all cycles back to Shafrov's rules, those base pairing rules. When we initially started teasing out the structure of DNA, that was one thing that he ended up contributing data to back in the 40s. All right. Regardless, you always had the same number of purines to pyrimidines. So if I told you you had 100 base pairs and 40 of them were purines, then you know that there also has to be 40 pyrimidines. Does that make sense? Um, Math doesn't work out quite that well, but you get the idea, right? So you have to have the same number in each thing. If I have 40 thymines, then you have to have 40 adenines. That's kind of where I was going with this. All right, but overall, 100 base pairs, 50 of them have to be purines, 50 of them have to be pyrimidines, which means if I know 40 thymines, 40 adenines, that means the other 20 have to be made up of cytosine and guanine. So you can backtrack and figure out, so long as you have one piece of information, you can figure out the rest. So when we look at the collective structure of DNA, right, this is a double-stranded molecule. It's a double helix. And so those two strands, right, are held together, connected by those complementary base pairs, the A's and T's, the C's and G's, like rungs in a ladder, right? Kind of think of this as a twisted ladder. And so when I look at what's holding those rungs together, right, these are your hydrogen bonds, okay? In addition, you then have that sugar phosphate backbone, so that alternating sugar to phosphate. And so when I look at that, there's also special bonds holding that together. And so when I look from every sugar to phosphate that is going on from one molecule to the next, all right, we're dealing with these phosphodiester bonds, okay? So when I look here, right, this would be a phosphodiester bond, okay? Um, and so you have those alternating bonds going on there as well. Other thing to keep in mind is that these strands don't run identically to each other. In fact, they run what we call anti-parallel. So when I look at the end strand of one molecule, it's your 5' end, which would be your free phosphate. So if I came up here, this would be a free phosphate. If I look up here, this would be that 5' end. That would then run down to a 3' end, which would be your free sugar. Okay, so if I were to look at this strand here, I'd have a sugar here running all the way down to a free phosphate. The other strand is the opposite, so I've got a free phosphate running all the way down to a free sugar. Where do those 3' and 5' come back to? Well, if you come back to the structure here, take a look at the carbons. They're numbered, right? So when we talk about numbering carbons, we're numbering the carbons from that sugar. So I've got a 1' carbon here, 2' carbon here, 3' carbon here, which is where we're giving that bonding connection to that is absent in that 3' end, up to the free phosphate, which is attached to the 5' carbon in that sugar molecule. Keep that 3' and 5' in your head, because the fact that these strands run anti-parallel to each other is going to complicate the way that DNA replication has to work. Because the enzymes that are necessary for the building of these molecules and the way that those nucleotides have to plug into each other as I build a polymer of nucleotides can only happen in one direction. And so when we start throwing out lagging strand and leading strand and the way that these strands have to get created or copied, that 5', 3' concept is going to come back. This one's just another way to take a look at this molecule. We often look at very much just those sort of skeletal structures of DNA, but this gives you a little bit better idea as to what the actual space is. It's a computer space filling model where you can see how big those groups and atoms are and how they kind of all fit together. It's a much more dense molecule than it kind of maybe appears from that more traditional skeletal ladder structure that you're typically used to seeing. So it's just another way to take a look at that mouth.